to our seminars, our monthly seminar series. I'm particularly delighted that Susan Ellenberg has come to speak to us today. We were just, for those who you heard earlier, Susan and I were reminiscing. We actually met for the first time in the late 1980s at the peak of the AIDS epidemic when she was working for NIAID, and Kurt and I were funded to begin what became the Studies of the Ocular Complications of AIDS, or SOCA network. And it was a unique time both for clinical research and in the AIDS epidemic, things that have changed dramatically with effective antiretroviral therapy. Susan's had a very distinguished career. She got her undergraduate degree from Radcliffe and her PhD in biostatistics from George Washington. She's worked at the FDA, at NIAID, and she has for a long time now been at the Perelman School of Medicine at the University of Pennsylvania, where she is now an emerita professor, having recently converted from full-time to emerita. And she is going to talk about designing trials of new vaccines, lessons learned from recent infectious disease outbreaks. Thank you, Susan.
Thank you, Doug. So I'm going to start by doing a little bit of a primer on evaluation of vaccines generally. There are some similarities to drugs and some major differences. So new vaccines are designed to elicit an immune response against a virus, a bacterium, or a parasite. They'll typically be tested in one or more animal models before exposing humans, looking for immune responses. Occasionally, there will be, they will look to see whether there's protection of disease in animals if the animal model is appropriate. So if you see the desired immune response in animals, there's no worrisome safety issues, and there are no manufacturing issues. Manufacturing is a big issue in vaccine development. Those of you who are aware of, I'm sure most of you don't remember, but you're aware of what happened with the first polio vaccine, the Cutter incident, where manufacturing deficiencies initially caused cases of paralytic polio in some vaccine recipients. So vaccine manufacturing is looked at very, very carefully. But once all that's okay, the studies will proceed to humans to look for initial safety and immune response data. The FDA will work very closely with the manufacturer, looking at the protocol, agreeing that based on all the animal data that it's safe to proceed to human testing. Often different dose levels will be tested in phase 1. And if that's promising, the next stage will study larger number of subjects and will typically be a randomized study comparing outcomes in people who get the vaccine versus a placebo or another vaccine. Some of these phase 3 trials are randomized or use an imbalanced randomization scheme, 2-to-1 or even 3-to-1, because of the anxiety to get as much safety data as possible on the vaccine. You know, those of you who are hearing this talk understand about randomization and why we do it. I will tell you, when I joined the FDA in the early 1990s, there were a lot of people there who still thought it would be okay to evaluate a new vaccine without a randomized trial. I think that thinking is pretty much gone, except for some very, very difficult circumstances. So trial size depends on the expected event rate, the desired or the expected level of benefit, and occasionally concern about rare side effects if there's some reason to expect or to be worried about a particular side effect. Now, the efficacy standard is somewhat different from that for drugs, where you're looking for a p-value less than 0.05, basically assuring that there's at least some benefit. Because vaccines are given to large numbers of healthy people, the efficacy standards tend to be higher. So it's not enough to just show that the confidence interval is above 0. You want to be very sure that the vaccine has more efficacy than that. So if you have a vaccine where you expect very high efficacy, you might want that lower confidence interval to be above 50%. In recent vaccine studies, it's been determined, for the COVID vaccines in particular, that the minimum level of efficacy that would be acceptable would be 50%, and that the lower confidence interval had to be above 30%. So that's something different. And the considerations for how this is looked at is the severity of the disease to be prevented, how urgent the need is, and any anticipated safety issues. So some special issues for vaccine trials. There's a sort of a different philosophy about vaccine trials. Safety is obviously very important. Vaccines are given to healthy people. Not only that, for a drug, if you're taking it for a medical condition, you know whether the drug is doing something for you. If you're taking it because you've got a runny nose all the time and you take an antihistamine, you know whether it's helping you. You're not sneezing as much. And obviously for more serious disease, there are ways to see whether the drug is helping you. But a vaccine is intended to prevent something that might never happen, right? Because if you get vaccinated, you don't know whether you would have gotten sick if you hadn't gotten the vaccine. So you can't determine on an individual basis whether the vaccine is helping you. Successful vaccine trials are often followed by large observational studies to identify less common side effects, something that it happens sometimes with drugs, but not too often. Some interesting methodological considerations. Intent to treat has never really taken hold among vaccine clinical trialists. Sorry to say, they just don't believe in this. Most vaccines are given as a 2 or 3 dose series, and they argue that typically, and it's true, typically a very high percentage of participants receive all the doses and have the right amount of follow-up. So intention to treat usually wouldn't differ from a completers analysis. In all my years at the FDA, I was never able to persuade the regulators that intention to treat should be the primary, but they do look at it now. I don't think they didn't used to even look at intention to treat. Despite the concerns about safety, many vaccine trials have been quite small. If you have a common disease, you don't need a large population to and with expected efficacy to be high. You don't need a lot of people to show that to demonstrate that the vaccine's effective. And as I said, the additional safety data is often obtained in observational cohorts. A big issue with vaccines is trying to find correlates of protection. So that's the vaccine version of surrogate endpoints. These are the immune responses to vaccines. Sometimes it's very clear what immune response is causing protection from disease. In other cases, there's typically a number of different types of immune responses, and it's not clear which ones are the ones that actually cause protection. And it's important to find out, because often people want to test a new vaccine formulation. Maybe it's more efficient, or you're getting rid of a component that may be causing some side effects, or especially when vaccines are tested in combination. You can no longer do clinical efficacy studies because you pretty much, the disease has become quite rare because of the use of vaccines. So if you are testing combination, you've really got to look and see whether the immune responses have not been altered when you give the vaccines in combination. Another important difference with vaccines is the concept of herd immunity. Vaccines affect more than the person vaccinated, because people who are protected from infection or disease are less likely to transmit infection to others. And when the vaccine coverage is high, the population risk can be greatly reduced. We've even managed to completely eradicate smallpox. We were very close to eradicating polio, didn't quite get there. But when coverage declines, the risk of infection goes up. We've had measles outbreaks in recent years, and even more recently, we've seen cases of polio. And this is what happens when when people stop getting vaccinated. There are vaccine trials that are designed to capture the indirect as well as the direct effects of vaccine. Don't have time to go into those details, but a very nice paper by Betts Halloran, published about 10 years ago, talks about some of those methods. Another type of study that is sometimes done with vaccines is a challenge study, where an individual receives the investigational vaccine and then they're exposed to the pathogen. This is can be done in animals if there's a good animal model for human disease. It can be done in humans only if there's a known treatment that would cure or mitigate disease in those who become infected. And challenge studies have been done in malaria, typhoid, cholera. Again, I wasn't aware of challenge studies, and I remember when I was at the FDA hearing about a study of a new malaria vaccine where people willingly put their arms into a large tube of mosquitoes and allowed themselves to be malaria-infected mosquitoes and allowed themselves to get malaria. And it was a particular strain of malaria that was known to be curable by disease. So these kinds of studies obviously can be controversial. They were talked about early in the COVID-19 epidemic before people assumed that it would take a long time to develop vaccines, and people thought, well, this would be, you might find a new vaccine in a shorter period of time if we use challenge studies. It was controversial because there was in fact no known cure or even known treatments that would clearly mitigate the disease. They were implemented. It turned out they weren't needed, but there was some additional information that was gained from those studies about immunogenicity. So now I'm going to move to talking about some of the issues that arose with the development of vaccines for Ebola and COVID-19. So, oops, typo there, 2014 and 2015, there was an Ebola virus disease outbreak in West Africa, the countries of Guinea, Sierra Leone, and Liberia. This was the largest Ebola outbreak ever. I think the first Ebola outbreak that was identified was back in the 1970s, and they tended to be small. In this outbreak, over 28,000 individuals were infected, 11,000 deaths. There were no known effective treatments or vaccines. It was a horrible disease, highly infectious, high fatality rate, very limited healthcare facilities in areas of outbreaks. It was important to be able to effectively communicate with community leaders to plan and conduct the trials. Most of the clinical trials that were established for Ebola were on drug treatments, but vaccine candidates were tested also. So this shows the time course of the epidemic in West Africa, and it was a very rapid peak and then a very rapid decline. So the cases occurred within a period of just a small number of months, less than a year, from the start of the epidemic in about in early 2014, where it really started to peak in about the middle of 2014, and was waning by early 2015. So the good news for vaccine development was that Merck had been working on an Ebola vaccine, and it was ready to go into clinical testing. So several randomized trials were actually initiated. One was sponsored by NIH. It was a classic individual double-blind placebo-controlled randomized trial in the general population. Another randomized trial, I think this was run by the CDC, had a delayed vaccination arm as a control with an 18 to 24-week delay in high-risk individuals such as healthcare workers. And third was a ring design. This was modeled, for those of you who know something about vaccination history, ring design was modeled on the approach to smallpox eradication, which was done in the 1960s and 70s, but it had never before been used to evaluate a new vaccine. It was used to actually control the spread of smallpox. Now, the bad news is the fact that the epidemic spiked so rapidly. It was not easy to get these trials started. There were a lot of issues in negotiations with the affected countries, stressing the importance of isolating active cases, interference with traditional burial practices and other traditional practices that made it difficult to keep people isolated and to keep them away from getting infected, and then the acceptability of placebo controls. So investigators were unable to actually initiate the clinical trials until the outbreak was almost over. And you can see that on this slide. This is the same slide I showed you before, the first identification of Ebola, the rapid spike, and the trials weren't initiated until over here, until the epidemic was almost over. So let's talk about the ring trial. The way this was designed was a ring was defined around an identified case. So there was a case, then all individuals who had direct contact with that case, and then all individuals who had direct contact with the case's direct contacts defined a ring. And then as soon as a ring was defined, each new ring was randomly assigned to either immediate vaccination or vaccination after 21 days. So it was a type of cluster randomized design, but with the clusters defined as time progressed around identified cases. So this design did maintain an element of randomization while ensuring that all participants would eventually get the vaccine, which was an important issue in negotiating with the countries. This trial was the only vaccine trial to report efficacy results. It was actually stopped early by their DSMB at a time when there were no cases in those immediately vaccinated and 16 cases in those in the delayed clusters. Actually, I think I misspoke. I think this was the final results. I think it was stopped before there were as many as 16 cases in the delayed clusters. Now, there were some issues in interpreting these results. I mentioned intention to treat before. About a third of those in the immediate clusters actually never got vaccinated. So this was a much higher proportion than you usually saw in a vaccine clinical trial. And these were excluded from the primary analysis. So the 0 versus 16 excluded those people who didn't get vaccinated in the immediate clusters. When you included them, then there were 7 cases in the immediate clusters and 16 in the delayed clusters. So this was suggestive, but it didn't reach statistical significance. Nevertheless, the vaccine did get approved by the FDA, and it has been deployed in countries experiencing outbreaks. And I've given the citation to the major publication. The 2 other randomized trials couldn't assess efficacy because the epidemic had waned, but they did study almost 10,000 people using the same vaccine as in the ring trial. So they did contribute important safety and immunogenicity data. So a major lesson from this is to ensure definitive conclusions about effects of medical interventions during outbreaks. You may need to evaluate treatments over multiple outbreaks. And some of us who were working with the FDA published a paper about this a couple of years ago. None of the trials of Ebola treatments studied during the West African outbreak came to definitive conclusions, and there wouldn't have been a definitive vaccine answer either if the ring design hadn't been implemented. Now, this is challenging. The idea is, okay, you didn't get as much data as you needed. So now we hold tight, we don't publish the results, and we wait till the next outbreak and continue the study. So you can see that would be challenging. There was when there was an outbreak in the Congo, not too long after this, the treatments that looked positive, even though it wasn't quite definitive, were used. Now there's a new Ebola outbreak right now in Uganda, but it's a different strain from the earlier outbreak. And they know that the vaccine, the Merck vaccine, will not work against this strain. But there are several vaccine candidates for this strain, and they are being studied using the same ring design as in the earlier outbreak. So remains to be seen whether those will prove as effective as the one in West Africa. So now I want to move on to COVID-19, which is different from Ebola in many, many ways. The outbreak was global, not limited geographically, and actually affected more developed countries with good health facilities, didn't have the same problems as you had with Ebola in West Africa. Most cases of disease were mild or moderate, and in fact, many were asymptomatic. The mortality rate was non-trivial, but certainly much lower than for Ebola. There was a community recognition that people needed to be isolated. We didn't have to deal with cultural problems like we did with Ebola. There were no difficulties in communicating with the public. Everybody has TVs, everybody has phones. That was not the case in Africa, and it was often difficult to communicate with people in rural communities there. But one important similarity was the readiness for vaccine trials. With Ebola, it was not a new infection, and companies had been working on a vaccine, were ready to put it to the test. With COVID, something rather astounding many of us, the virus was rapidly sequenced, and the years of prior work on mRNA vaccines meant that they were really ready to go in terms of testing, as we know. So multiple companies embarked on development programs, not all using the mRNA technology, some using the more traditional vaccine approaches. And the NIH partnered with 5 companies to carry out the required trials. The FDA, together with its advisors, determined that to be successful, the estimate of protective effect would have to be at least 50%, with a very high probability that the effect was at least 30%, and required at least 2 months of safety data on at least half the subjects. Now, these trials were quite large in order to get reliable answers as quickly as possible. It's important to remember that these trials focused on illness, not infection alone. So an outcome in these trials was some level of symptoms plus documented infection. They also looked separately at severe illness and death, in addition to the mild or moderate symptoms, and with special attention in subgroups of older people and people with pre-existing conditions that might make them more susceptible to bad outcomes. So there was a huge pressure to get data faster, but great concerns about safety, given that this vaccine would be given, or these vaccines would be given to potentially billions of people. So it was very important to look for rare but serious events. Now, we know that during the trial, we're going to see some bad events by chance. The trials had specific thresholds. They had to have a certain proportion of people over the age of 60 and people with comorbidity. So these people were going to have bad events whether they got COVID or not. And so this was a big challenge for the DSMB to watch and see whether you were having more of these bad events early on the vaccine arm. So as you might expect, there was an independent group of researchers, the DSMB, that was monitoring the data from these trials. This was the only group that saw the comparative data. Data for each trial were reviewed monthly, with any serious adverse events sent to the board in real time. And so this board, on which I served, met via Zoom almost every week, given that there were multiple trials. And after each meeting, recommendations were made to an oversight group, which was not only the NIH, but also BARDA, the Biomedical Advanced Research and Development Authority, and the vaccine manufacturer. The DSMB had to pay particular attention to serious events, particularly deaths, which was difficult early in the trials, especially since many older people entered the trials early. There was particular attention to rare outcomes that have been seen with other vaccines, like Guillain-Barré syndrome and Bell's palsy. The trials were completed very quickly. People were breaking down the door to have a chance to receive a vaccine, and the high rate of transmission meant that cases came in very rapidly. The board included clinicians, epidemiologists, virologists, immunologists, and statisticians. They came from academic agencies, NGOs, government agencies, and from the US, UK, Brazil, and South Africa. And this board, we reviewed the emerging data for each trial until all the data had been made available to the sponsoring company, to NIH and the FDA. And we finally had our last board meeting this past September. So as I mentioned, the interim reviews focused on emerging safety data and also consideration of whether the trials could be stopped early if the efficacy was very strong. A big issue in monitoring was trial conduct, whether there were reporting delays, missing values, inadequate representation of important subgroups. This was something we looked at very carefully. We wanted to make sure that the trials at the end of the day, would be credible. There were some worrisome events identified early on, the thrombi, blood clots, myocarditis. These events also occurred in those with COVID-19. So initially it wasn't clear whether they were truly caused by the vaccine, but they certainly were cause for concern. And even if they proved to be a rare side effect, there was the risk-benefit issue. If a vaccine is highly effective, does that benefit outweigh the very small risk of a serious adverse effect? Ultimately, the blood clots and myocarditis were thought to be likely rare effects, but fortunately almost always treatable. Most serious events did occur at similar rates in the vaccine and placebo groups. Another thing that was taken account of in monitoring these trials was the possibility of disease enhancement. This means people who got the vaccine, if they did get infected, would have more serious disease. And we have seen this in the past. Probably the most well-known example was a candidate vaccine for respiratory syncytial virus back in the 1960s, where it was shown that not only did the vaccine not protect children from infection with RSV, the children who got infected had more severe disease than children in the control arm who received a flu vaccine. This was such a huge setback for RSV vaccine development that we still do not have RSV vaccines. And as many of you know, we're having a huge outbreak now of RSV. The children's hospitals are flooded with cases. A new study of RSV vaccine in older adults has just been reported. I don't think it's out in a paper yet that looks positive. So there's some hope there. For COVID vaccines, we looked at the severity of cases observed in those who got the vaccine versus those who got the placebo, and formal boundaries were set up for excess severity among vaccinees. But fortunately, this did not turn out to be a problem for any of the vaccines that we studied. Now, there are many challenges with the ongoing studies of COVID vaccine candidates. The earliest vaccine candidates, Moderna and Pfizer, we were not monitoring Pfizer, that was done separately, but these were authorized while the trials of other candidates were in process. So many participants dropped out to receive authorized vaccine, as they certainly should have. The ongoing trials had difficulty recruiting older people who were the first in line to get the authorized vaccines. Exclusions, and initially people who had already been infected were excluded from the trials, and that caused an increase in exclusions, so more problems with recruitment. And so the conduct and analysis of these later trials became more complicated. Now a crossover design was put forward by Dean Follman and colleagues, and some trials implemented this crossover plan, which involved those who originally got, they would randomize. I'm sorry, those who originally got vaccine would get placebo and vice versa, with no one being told what they got the first time. So everybody going into the trial would know they would eventually get a vaccine, but they wouldn't know which one they got first. And this kept people in trials and increased information about vaccine efficacy. And a couple of trials did use this design. Another complication, of course, was the emergence of new variants. The first few authorized vaccines were developed based on the initial viral variant. the vaccine candidates that got a later start were evaluated in the setting of newer variants, they still showed efficacy and a high level of protection against the most serious diseases. So that was good news. There were many challenges for the DSMB in this trial. Despite NIH efforts to make the trials identically designed, the definitions of the primary outcome differed slightly. The assay requirements for proof of infection, I'm not sure that all of them required central assays. The list of symptoms and the level of severity required was not 100% consistent across trials. Probably the biggest issue was that each trial had its own data management and statistical group. So the ports had varied formats and contents. Now, I have served on data monitoring committees, DSMBs, for trials trials run by cooperative groups where you monitor multiple trials that are run by this group, but it's all the same statistical and data management center. And that makes a world of difference. The quality of these reports varied greatly. There was so much urgency, it was difficult. The independent statisticians frequently were unable to answer questions or provide additional data, to the point that we requested that companies included an unblinded clinician to work with the reporting statistician to help us understand some of the emerging data. Another issue was that the adjudication of the primary outcome often lagged substantially. So we were never, we're often not quite sure where we were in terms of the number of outcomes that were going to actually be counted. It's important to remember that the primary trial outcome was symptomatic disease, not just infection. And they were only tested. Not everybody wasn't tested throughout the trial. They were only tested after the second dose if they developed symptoms. So the trial results therefore reported protection from symptomatic disease. And I think a lot of the public didn't really understand this, that you were not protected against actual infection. I'll talk a little bit more about that later. So there were certainly public concerns that emerged. Most of us were desperate to get vaccinated once vaccines were available, but some remained skeptical. The mRNA technology was new. The vaccines were developed so fast, could really be sure that they were safe? And there was concern about the expected moderate side effects. People knew it was going to, you might run a fever for a couple of days, you might be fatigued for a few days. So even forgetting the possibility of serious side effects, people were worried about maybe missing a few days of work. So there was some hesitation about getting the vaccine. And then the reporting of the newly identified adverse events, such as myocarditis, just enhanced concerns. And finally, the downplaying of the seriousness of COVID-19 by politicians. Some politicians supported skepticism about the need for vaccines. Then there were a lot of people trying to do analysis of vaccine data that they had available that purported to show lack of efficacy or harm. Those analyses were faulty. There were many people following those kinds of analyses to try and rebut them. But certainly reports of breakthrough COVID-19 cases among those already somewhat skeptical just raised more doubt. So I want to digress and talk a little bit about vaccine resistance. There's a long history of vaccine resistance. As some of you know, Benjamin Franklin refused the smallpox vaccination, which had been developed during his time for his child, who then later died of smallpox. But more recent issues, pertussis vaccination led to concerns about relation of the vaccine and sudden infant death syndrome, also neurological problems. There were organizations founded that promoted vaccine misinformation back in the 1980s. And then, of course, there was the measles vaccine and autism scare in the 1990s. So these issues were spurred by individual reports of bad events occurring in children shortly after vaccination. So you have to think about the math here. 4 million live births a year in the US, vaccinations given multiple times during the first year of life. Mortality in the first year of life is higher than in any other years until you get to the mid-60s. 5.6 deaths per 1,000 live births. That's data from CDC from 2019. Now, about half of these are due to congenital malformations or complications of birth or other issues with newborns. So if you take those away, so you have 2.8 deaths per 1,000 live births, that means about over 11,000 infant deaths a year. Now, infant vaccination schedules call for vaccines at birth and at 2, 4, 6, and 12 months. Some of these deaths, more than a handful, will occur within a week of a child having been vaccinated by chance alone. Vaccines are not going to protect you against every bad thing that could happen. But a parent who has their child vaccinated and then something bad happens to the child within a few days after the vaccine, many of those parents are going to be convinced that the vaccine killed their child. And of course, it's not just deaths. There are many other medical conditions that arise in the first year of life. Now, some types of outcomes are readily attributable to a vaccine. Fever, chills, swelling at the vaccine site. Nobody worries that those are coincidental events. Also, somewhat more serious hypersensitivity reactions can happen. Most serious outcomes have no plausible relation to a vaccine, but if someone gets vaccinated and then has a heart attack the next day, they may well suspect that the vaccination was the cause. The FDA and the CDC have a reporting system that's full of reports of things that happened after vaccination. So, and that's called the Vaccine Adverse Event Reporting System. Some of you may be familiar with this. Anyone who experiences an adverse health event after being vaccinated can report it to VAERS. This database is publicly accessible. It's not edited. Whatever's reported goes right into the database. If a rare but serious event were associated with a newly deployed vaccine, this database, however messy it is, might actually be the quickest way to identify it. We did, this database was essential in identifying a rare intestinal problem associated with the first rotavirus vaccine. But reports being in the database certainly cannot be assumed to have been caused by vaccines. So some examples: a 70-year-old man receives his yearly flu vaccine, has a fatal heart attack a month later. A 30-year-old woman receives the HPV vaccine, is diagnosed with multiple sclerosis 6 months later. A 6-year-old child receives several vaccines, 2 months later is diagnosed with leukemia. Now, the VAERS is full of events like these. And so these data have been used to attack the COVID-19 vaccines. The first priority for vaccination was older people, very elderly, especially those in nursing homes. Some of you may remember. So these people, most of them had other health problems, had limited life expectancy. Now, these deaths were reported to VAERS, and they were taken up by anti-vaccination bloggers and commentators on conservative news outlets as evidence that the vaccines were actually killing people. Other public health reporting has been misinterpreted to create concerns about vaccine efficacy. And this is an interesting example that I wanted to share with you. So Israel was very good about collecting a lot of data. And they reported in the summer of 2021, they reported 515 individuals were hospitalized with severe COVID-19 infection. Okay, so severe, like hospitalized. Of these, more than half had been vaccinated. Now, when this was reported, there was some initial panic. Does this mean that the vaccines are not working? But most of the people were vaccinated. Almost 80% of the population was fully vaccinated. Fewer than 20% remained unvaccinated. So when you look at the number of cases, yes, more than half the cases were in vaccinated groups, but it represented only 5.3 cases per 100,000 versus 16.4 cases per 100,000 in those who were unvaccinated. So that was about 67% efficacy. So, all right, 67% efficacy, it's not nothing, but it's lower than the initial estimates. Was this evidence at least of waning efficacy? But it was recognized that you had to look at the data by age because more older people get vaccinated and older people are more likely to get severe disease if infected. And when you look at when you look at it by age, you can see that those under the age of 50, more of the cases were actually in those who weren't vaccinated, and the efficacy versus severe disease was over 90%. If you look at those over the age of 50, more of those were fully vaccinated. But again, the cases, the estimated efficacy was 85%. So when you looked at it by age, you can see that there was not waning efficacy. It was still, the vaccines were still very effective. And this is an example of Simpson's paradox. Some of you will recognize this. This kind of anomaly can happen when both the outcome and the risk factor are much higher in one group than in another. In this case, older people were more likely to be vaccinated, and if they were infected, were more likely to have serious disease. So this example illustrates the complexities of interpreting the emerging data on COVID-19 cases. But the anti-vax bloggers were very hard at work, and my colleague Jeff Morris at Penn maintains a blog, which I've given you the link here, who has been busy looking at all of these claims and refuting them. So it's pretty interesting. So just to conclude, making a few remarks, vaccine trials do raise a number of issues that don't tend to arise in therapeutic trials. And there were certainly more special issues that arose for vaccines developed during infectious disease outbreaks. Following the West Africa Ebola experience, WHO convened a blueprint committee as part of their research and development program to consider optimal designs to be used in future outbreaks. And I'm part of this group. A number of diseases are under consideration: Marburg virus disease, Lassa fever, Nipah virus disease, several others. And the big issue is the potential need to collect data over 2 or more outbreaks before data are definitive. Now, in this country, we certainly have examples where you have a seasonal disease and you have to collect data over 2 or more seasons before you're going to have a definitive result. But of course, seasons are predictable and outbreaks are not predictable. So I think this is going to be challenging to get people to do this. Certainly, we still need vaccines for some diseases common in developed countries. RSV, as I mentioned, Lyme disease. We had a Lyme disease vaccine. The manufacturer took it off the market because people believed without much good basis, we at the FDA thought that it was causing problems. HIV still doesn't have a vaccine. Group B strep, there are others. But increasing vaccine hesitancy may make trials harder to do and are likely to limit uptake of vaccines with proven efficacy and safety. I think public health officials are very concerned now about vaccine coverage. I think the issues with the COVID-19 vaccines have caused people to be more anxious about vaccines that they didn't worry about for many years, the standard childhood vaccine. So these are worrisome days for public health officials and public health in general. And I will stop there and see if there are any questions or comments. I know some of you in the audience are very knowledgeable and have lots of experience with vaccine trials and may have other things to offer. Looking at you, Larry.
Yeah, I'm here. All right. Hi, that was really comprehensive. A great introduction and covered a lot of stuff. I just have one question is about the COVID vaccines in particular. FDA doesn't seem to have worried much about the partial unblinding that takes place in especially the mRNA vaccine trials, where people who get the vaccine, they get a fair amount of reactogenicity. And so, they can actually very often guess that they got the active vaccine. And so they're partially unblinded and may change their behavior and may get more exposed or less exposed. I was just wondering if FDA has thought much about this. And I don't see anyone in these trials actually asking all the participants what we used to do for some other studies. Do you think you got the vaccine? In which case you can look for differentials and so forth.
Well, it was interesting because when we looked at the adverse event reports, there were a fair number of people who got the placebo who also reported fever, malaise, chills, some of the same kinds of things. You're certainly right that that people may have suspected that they got the vaccine. I don't know whether early in the epidemic, how much behavior would have changed. People were still very afraid of the virus, and if they did, then they would be more likely, maybe, to get infected. But in fact, the vaccines showed very high levels of efficacy. I mean, the first vaccines was in the neighborhood of 95%. So, as with many treatments, not just vaccines, we know there's going to be partial unblinding due to side effects. Not much we can do about it. We can't do anything to make the placebo people more sick than they would otherwise have been. I don't think the ethicists would look kindly on that. So, but I think the high levels of efficacy diminished concerns about unblinding.
Great. By the way, Susan, absolutely excellent talk. I see Janet's hand up.
Oh, yeah. I wanted to ask two questions. One was kind of a political question. I've always wondered that the DSMB meeting was the weekend after the election. And right after the election in 2020, then these very positive results were released, and I'm just wondering if someone was thoughtful about that. That might have been a good idea. And my second really unrelated question is for the ring design. There seems to be kind of that would be have a lot of ethical issues about the logistics of implementing that kind of design.
So politically, I know that there were some issues there where I think the previous administration may have felt that they didn't get credit for the vaccines but the results were announced when the results were available. That's all I can tell you. There was absolutely no attempt. People were concerned about getting good information out as quickly as possible, not about who was going to get credit for it. So
I wasn't implying that, but the meeting was indeed scheduled for the weekend after the election that the DSMB met. And
well, these meetings were on a regular schedule. They were scheduled well in advance. I mean, like I said, the vaccines were looked at every month. Sometimes we had ad hoc meetings if something special had arisen, but the meeting schedule was set.
Okay, good to know. I mean, it's just sort of like you wonder about these things sometimes. If people are thoughtful about them, or sometimes the chips fall where they should.
I was on the DSMB. The DSMB didn't set the meeting times. Those were set by the by the NIH and the companies and BARDA. So I can't really speak for them, but I know these meetings were held very regularly. Now your second question was
about the ring design, about it.
So what did you think? What was the ethical concern that you had?
Well, that if you were around a case, then who consents the people who happen to be around the case and within 2 degrees of separation. It would seem...
there were people conducting the trial who did do informed consent. I mean, these trials were done with informed consent.
Okay. But was it a community consented ahead of time, or was it as cases were detected? Then the
ring was defined by the identification of a case. So once a ring was defined, then the first thing you had to identify the direct contacts of the case, and then the direct contacts of the direct contacts, and then those people would be consented. Now, certainly the community, I mean, there was a lot of negotiation, as I said, with the with the political leadership of the countries about doing this study. So, and there were lots of discussion with community leaders. So the community leaders were aware and had to agree, but the individuals were also consented.
Oh, okay. I just think it might be logistically challenging in the US. But it was fascinating to hear about a new design. Thanks.
Yeah, I doubt that this design would ever be used in the US.
Yeah. And
It's not anybody's first choice of design. Okay, there are a lot of flaws with this design, and if the vaccine hadn't been so effective, I think we really would not have known what to make of it. And even with the looking as effective as it was, there were some concerns because of the fact that, you know, a third of the people who were in the immediate vaccine ring didn't get vaccinated, and some of them got infected. And, you know, there are others. You can imagine all the difficulties in conducting trials in this environment. So I don't, but it was the only kind of trial that I think people saw that this kind of trial might actually get answers, even though the epidemic was waning, and they were right.
well, great talk. Thank you.
So I see 3 more hands up. Zhang Ji, I think, has her hand up.
Yes. Hi, Dr. Ellenberg. Thank you so much for your talk. I have a question regarding potential between group contamination in vaccine trials. We have one group vaccinated and one group unvaccinated. And if those participants come from like a small village, like in the ring design, they are around the same index person. So those vaccinated go out there and they may cause some herd immunity, right? So that may result in a conservative estimate, which I don't think is necessarily a bad thing, but I just wonder if, you know, this is an issue that's considered in vaccine trials and if there are any measures to deal with it.
Well, so in the ring trial that was done, it was a cluster design. So a ring was a cluster. So everybody in the ring was either got immediate vaccination or the delayed vaccination. So if you had individual randomization within the ring, then you would have had that issue. But in this particular, and that might actually be a better design and would actually allow you to get some estimates of the indirect effects. And again, that paper by Betz Halloran, I think, you know, talks about those kinds of designs. But in this ring trial, the whole cluster was randomized to either get the vaccine right away or not. So you didn't have people within a cluster, some vaccinated and some not.
Oh, thank you for that. I thought we like divide people into 2 parts and one part get vaccinated and the other didn't. So yeah, so we use cluster randomization to deal with the potential contamination. Yeah.
Yes.
Thank you.
So I think I see Nansu has to talk, a question rather.
Yeah, that's hi, that's okay. Let's have Jingqi and Leon first. Yeah, that's okay. Thank you.
Okay. I can't see all of the questions. If someone else has one, please just go ahead and ask.
Hi, Dr. Ellenberg. Thanks for a great talk. Can people hear me okay?
Yeah.
Yes.
Great. A question I have is, with age being such a strong prognostic covariate in COVID, was there any, I'm wondering how, if there was sort of consideration of conditional versus marginal estimands for the primary efficacy outcome, and how, if at all, that was considered?
Well, certainly we looked separately at people in different age groups to see you know, to see what the efficacy was. Now, the efficacy was high everywhere. So we really, you know, we really didn't have to go through a lot of complicated, more complicated, more sophisticated types of analyses. It was clear that the vaccine was effective in pretty much everybody.
Makes sense. Thank you.
All right, maybe one more. I have something else I have to jump to. It looks like it's just about 9:30. So I see a hand up. Dr. Zhan?
Hi, this is Jingxi, and I have a question about the side effects, the long-term side effects. So one of the big concerns of the new technology RNA vaccine is its like long-term adverse events. And instead of like, other than wait and see in the future, I wonder if there's anything we can do now or have been done in the trial stage to explore this concern. For example, like longer post-trial monitoring period or something?
Yeah, so the companies were all required to follow the people in the trials for 2 years. So we haven't seen 2-year data yet, but those will come out. Now there's not, I don't know that there's evidence of long-term side effects from vaccines. That's why initially approval was based on at least half the people in the trial had to be followed for at least 2 months, because for the most part, any side effects of vaccines are observed within the first 60 days after vaccination. But you're right, with the mRNA technology, you know, it's possible. You couldn't rule it out. But the companies are monitoring the people who were in the trials for up to 2 years. So we should have some information about that.
Okay, thank you.
Okay.
So, Susan, I think your time is now up. I know you have to run off. Thank you for a really excellent seminar. We very much appreciate it.
You're very welcome. I enjoyed it. Bye all.
Thank you.